such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
All right, so that's just a review of what you learned.
Are those available to us online? Or?
They actually, you can find a lot of good ones on YouTube, and YouTube does have Campbell's version of active transport, yes. But also, if you have Mastering Biology, that's what I do, is I just go to the Mastering Biology site.
Like the access code?
Yeah, there is no, yeah, there, and students will say, well, it asks you for a course ID. We don't have a course ID in this, so you have to do it as an individual, and you can still do it, but I don't know the process of that. But yeah, you can see these all over the place. They're good at bringing that stuff to life. Okay, so we're finished with membranes, and now we're starting with cell division. And you already did the lab today. So the lab is kind of like an overview of the lecture. The lecture is going to be more detailed, but the lab is a nice overview of the lecture. And I wish this thing worked, but it doesn't. Okay, so here's a question, just kind of a rhetorical question, but conceptually, you need to understand why the genetic code is important and why it must be passed accurately from cell to cell. Well, remember part of that diagram that you had to draw today. DNA replicates and has info for its own replication. What's that? Did somebody say something? Oh, I'm hearing things. You guys should have said, you should have said something, and you can mess with me easy, okay? The information on the DNA is transcribed onto a molecule of RNA, which is translated to a protein, Okay, remember that? And I'm going to stop there for this question. So why is the genetic code important and why must it be passed accurately from cell to cell? Well, everything we've worked with so far has to do with chemistry and molecules, right? Absolutely everything. Many of those molecules are proteins, and many of those proteins are enzymes. What does a protein's function depend on? No, what's it? Shape. A protein's function depends on its three-dimensional shape. Okay? So the three-dimensional shape has to be right. The only way the three-dimensional shape can be right is if the amino acid sequence is right. And the only way the amino acid sequence can be right is if the genetic code coding for that amino acid sequence is right. So that has to be passed accurately from cell to cell, because if we make a mistake, if the cell makes a mistake on a critical enzyme, it's curtains. Okay? So we're going to begin to look at how this genetic information is passed from cell to cell. Okay? And so the continuity of life is based on the reproduction of cells or cell division. Okay? So a question, why do cells divide? Well, there are different reasons depending on the organism. In unicellular organisms, the division of one cell produces an entirely new organism. Okay? So here's an amoeba right here. It divides, and we get two new amoebas. Okay? And there's a photo of an amoeba dividing there. I guess when the amoeba gets confused too, it wants to go in opposite directions, that could happen. I don't know. I don't think it would. But there's an amoeba dividing. Multicellular organisms also, the cells have to divide. Okay? First off, we all developed from a fertilized egg. And the only way we developed is during embryonic development is for the cells to divide. And then after we were born, to grow, the cells divide, okay? And they still divide. They keep dividing. So growth, development of a fertilized egg, and growth. Okay, and I don't like this right here. I'd rather have a better picture of of about the 32 or 64 cell stage. Okay, but I guess they wanted to keep it simple. Also, repair. Think about if you scrape your elbow or something like that, and the healing process starts, and once the bleeding is taken care of and things like that, then the cells start to begin to divide and replace the tissue that you tore away. So tissue renewal. Okay? Cell division. Most cell division results in daughter cells with identical genetic information. That means DNA, okay? So looking at this amoeba up here, these would be daughter cells, and they are genetically identical to each other and genetically identical to the original cell. Why is that? Because the DNA replicated, okay? The DNA replicated, and this cell got an entire complement of DNA, and this cell got an entire complement. So these are all genetically identical. And that's what mitosis produces, is genetically identical cells. So cells that are genetically identical are also what? What could you call them? What's that? No. The whole cell we're talking about. clones. Okay? Cells that are genetically identical are called clones. All right? Are the cells in your body clones? What's that? Which ones aren't? Actually, every cell in your body is a clone, except for sperm or egg. Okay? We'll get to that a little bit. Yeah. So every cell in your body has the exact same complement of DNA, except for sperm or egg. Every cell has the same set of instructions. So we'll just contemplate that a little bit because it's not readily obvious. Okay, it would be without biology, you would think that the brain cells were genetically different from liver cells. But things we know in biology now, we can explain why the cells have different functions in the same genetic code. So all the cells in your body are clones, except for sperm and egg. Okay? So they introduced that here, okay? A special type of division produces non-identical daughter cells that would be sperm and egg, also known as gametes. And that's meiosis, and we'll mention it here and there, but we'll get into it in detail later this semester in lecture. But these are the only cells in your body that are not genetically identical to the rest of the cells in your body. They will actually be unique. You've heard the term genome, okay? That means all the DNA in a cell, all right? The cell's genetic code is its genome. And every cell in your body has the exact same genome. Look, let me take that back a little bit. Red blood cells don't have a nucleus, okay? So they did have one when they were getting started, but they lost it. But you don't need to know that for me. You'll need to know. What's that?
I just have a question generally. So say like a skin cell of mine, it contains all the instructions needed for who I am, basically, and
You bet.
like what I've come to be in the body.
Yes. Yep.
It's pretty impressive.
It's absolutely amazing. Yeah. And all of that information is contained in that cell's DNA. The concept of information in and of itself is what blows my mind, because information is a non-material entity. And if we look, I mean, in science, the only thing there is materialism. Okay? Information in and of itself is non-material. So, and I'll get to that later when we get into DNA. Don't worry about that. It's a little bit of philosophical gobbledygook. Me trying to pretend I'm a smart guy. Okay. So a genome can consist of a single molecule, in other words, just a single chromosome, like in prokaryotic cells, or a number of molecules common in eukaryotic cells. How many chromosomes do humans have, by the way? What's that? 23? 23 pairs. 46 chromosomes in each cell. Okay? All right? So DNA molecules are packaged into chromosomes. All right? And humans have a characteristic number of 46. Now, so here's a cell here. The chromatin that was in this nucleus looked like this right here, kind of like this, but it has condensed into visible chromosomes. Okay? So chromatin and chromosomes are the same thing. They're both chromosomes. You just cannot see them in the form of chromatin. But they are there. And they are fully functional. They are working. In fact, they do most of their work in the form of chromatin. They only divide in the form of chromosomes. Okay? So each species has a characteristic number of chromosomes in each cell nucleus. All right? In humans, the characteristic number is 46. Does that mean no other species can have 46 chromosomes? No. Other species can have the same number of chromosomes. That just means if you're a human, if you have 45 or 47 chromosomes, there's usually something awry, okay, something wrong. So humans need to have 46 chromosomes. All right? I know you learned this term last week in your cell lab, somatic cells. Those are the non-reproductive cells. Somatic cells have 2 sets of chromosomes. Every cell in your body is a somatic cell except for sperm or egg. Those are obviously reproductive cells, gametes. Okay? So somatic cells, non-reproductive cells. Gametes, so we're mentioning them again here. They are the reproductive cells, sperm and egg. They only have half as many chromosomes. In other words, in a human sperm would contain 23 chromosomes, a human egg 23. Okay? They don't have 46. All other cells have 46 chromosomes. So eukaryotic cells consist of chromatin. Chromatin is the same thing as chromosomes, but in the chromatin form, it's a complex of DNA and protein that you can't see. It has to condense before you can see it, and it condenses for cell division. It doesn't condense so we can see it. We just happen to be able to see it during cell division. Okay? So, in preparation for cell division, the DNA is replicated, and it has to be, because there's no way that each cell will get the same complement of chromosomes as that first cell if the DNA wasn't replicated first. Okay? And we're still not looking at DNA replication yet. We're just getting a little closer to it. Now, each duplicated chromosome has 2 sister chromatids which separate during cell division. Okay, so let's say I'm just drawing one chromosome here, which is risky for me. Okay, I draw one. And what's going to happen before cell division, this is going to replicate. So I'm going to draw it like this. Okay? So now this chromosome is composed of 2 sister chromatids because the DNA has been replicated. And those chromatids are genetically identical to each other. Okay? How many chromosomes is this? Still 1. Okay? Because it is attached at the centromere. Okay? The centromere is the narrow waist of the duplicated chromosome. Okay? And that's where the 2 chromatids are closely attached at the centromere region. And here is the professional rendition of that. Notice to not embarrass them too much, I used purple just like they do here. Here's a nice electron micrograph of a pair of sister chromatids showing the centromere here, okay? So replication has already taken place. So in this organism here, we have one, we're just showing one chromosome it is going to be replicated into sister chromatids. That's still one chromosome, and eventually each new cell will get one of those chromosomes. Okay? This will happen with all 46 of your chromosomes. Obviously, we can't show all 46, right? Because that would be a mess. But this will happen with all 46 of yours. Okay?
So it still keeps that centromere even when they're not attached anymore?
What's that?
The centromere is always there even when
The
they're not attached?
centromere is actually this part and this part. Yes, okay? And then the centromere is the region at which they attach, but each chromosome has to have a centromere. Okay? Okay, eukaryotic cell division. So we're starting to get into that now. Consists of mitosis, that was today's lab, and that is nuclear division. So now you need to learn it. Okay, you didn't need to learn it during lab when I told you about it. Now you need to learn it. Mitosis is the division of the nucleus, and cytokinesis is the division of the cytoplasm. Okay? And the cytokinesis is going to differ in plant and animal cells. Okay? Now we are going to learn what is called the cell cycle. There is going to be a rather large fill-in-the-blank section on exam number 2 concerning the cell cycle. So you're going to need to learn the cell cycle to the level of detail that I lecture at. So it's going to be worth about, I think it's worth about 18 points. Okay? So the cell cycle consists of the M phase, which is the dividing phase, the mitotic phase, which is mitosis and cytokinesis. Okay? And we'll learn all the phases in there. And it consists of interphase, which is basically cell growth, DNA replication, and the cell getting ready to divide. Okay? The two main parts of the cell cycle. Mitosis and cytokinesis, the M phase, and interphase, cell growth, copying of chromosomes, so on. Okay? So now let's look at interphase first. Remember I described interphase and showed you pictures of interphase with that, that onion root tip slide. Interphase is about 90% of the cell cycle, and if you're looking at a slide with a bunch of cells on them and they're dividing, it's a slide to basically show cell division. The majority of those cells will be in interphase because it is about 90%. It occupies about 90% of the cell cycle. And there are 3 subphases of interphase. Okay? So you need to what the questions are going to look at. I will list an event that happens, and in a word bank, I will have all of the phases and subphases of mitosis. You will need to be able to put the right subphase with the right event. So let's look at the subphases of interphase. First subphase of interphase is the G1 phase, also known as the first gap. And you could call it first growth phase. Okay? The G1 phase is first growth. and what happens during the G1 phase, which you need to know. In other words, the lecture and the exam go somewhat beyond these slides, right? The details go beyond. The slides are the main thing, but sometimes the slide is inclusive. This is a case where it isn't, okay? So what happens during the G1 phase? The first thing would be proteins are synthesized. Okay? Proteins are synthesized during G1. Organelles are produced. Organelles are produced, and mitochondria and chloroplasts divide. Right? Proteins are synthesized, organelles are produced, mitochondria and chloroplasts divide. The second subphase of interphase is the S phase. The S phase of interphase. So they call that the synthesis phase. Well, synthesis of what? We had synthesis of proteins during G1. So what could be synthesized here? DNA. That's where DNA replication occurs, is the S phase. Okay? So DNA is synthesized during the S phase, and if DNA is synthesized, then that is where sister chromatids are formed. So going from here to here occurred during the S phase. Okay? That's where sister chromatids are formed, during the S phase. DNA is synthesized. Then you have the G2 phase, which is the second gap phase. And the G2 phase basically is the cell gets ready for cell division. There's more cell growth, and the cell gets ready for cell division. G2. So everything I said there is what you need to know about interphase. And here's just a nice little diagram of interphase right here. Well, the cell cycle in general shows interphase being 90% of the cell cycle. We have this to learn yet, the mitotic phase. But the interphase, here's G1, okay? The cell grows, first gap phase. Proteins are synthesized. Organelles are produced, mitochondria and chloroplasts divide. In the S phase, that's where the DNA is replicated. So sister chromatids are formed during the S phase. And then in the G2, the cell gets ready for cell division. Now, a question here. How much DNA is there in the G2 phase relative to the G1? Double, because the DNA was replicated here. Not double the number of chromosomes, double the amount of DNA, okay? DNA was replicated here, so this has twice the DNA that the cell had here, okay? So now we're going to look at mitosis, all right? And we're going to divide it into 4 phases that we talked about a little bit in lab: prophase, metaphase, anaphase, then telophase with cytokinesis. The one that I'm leaving out is prometaphase. I will count what the book says happens in prometaphase. I will count that as prophase. It's a late prophase. Okay? And this is basically showing all the phases, and we're going to blow these up and look at them and look at what happens during each phase. Okay? so the first phase is prophase and prometaphase. These are the same thing to me, okay? So if the book says prometaphase, make sure you write prophase. Probably won't have to write prophase because it'll be on the exam in the word bank. Prometaphase won't be there, okay? So what happens during prophase? One of the first things that happens is the nucleolus breaks down or disappears. The nucleolus disappears and the nuclear membrane breaks down. Okay? So the nucleolus will disappear and the nuclear membrane will break down. So here's prophase here, and that is 2. Here is G2 of interphase. Okay? See, it still has the nucleolus present in the nucleus in G2, and the nuclear membrane is intact. Well, in prophase, the nucleolus is gone, and the nuclear membrane breaks down. Okay? Another thing that happens during prophase is the chromatin condenses into observable chromosomes. Okay? The chromatin condenses into observable chromosomes. So you can see that here. If you look at G2, we see all this spaghetti stuff here. Okay? And we look at prophase, and we see that the chromosomes are condensed, they're visible, and they actually exist at this point as sister chromatids because this is G2, so we had to get through S first. So getting through S, then the DNA was replicated, then condensed, so we see the chromosomes in prophase. The centrosomes will organize something called the mitotic spindle. So I introduced the term spindle in lab. We're going to look at it now. You see these centrosomes here? By the way, taking away the possibility of plant cells and animal cells, taking that away, of plant cell walls and chloroplasts. Would this be a plant cell or an animal cell, and how can you tell? There's one way on here you can still tell.
Our animal cell has centrioles.
It's what?
It's an animal cell because it has centrioles.
That's right. It's an animal cell because it has centrioles in the centrosomes. Okay? So the mitotic spindle forms. So notice the centrosomes here. They're going to basically move toward opposite poles of the cell. And remember what a centrosome is. It's a microtubule organizing center. So spindle fibers are made out of microtubules. And that's a question in today's lab. Okay, one of those hidden ones in the reading. What are spindle fibers made of? Microtubules. Okay? So you can see the microtubules emanating out of the centrosomes here. So the mitotic spindle, the spindle is forming during prophase. Okay? So the spindle forms. And this is not prophase here, but it's just a really nice view of the spindle. Okay? So that's why I wanted to show you this. Just a great view of the spindle. But that's not prophase. Also during prophase, some spindle fibers attach to the kinetochore. What the heck is a kinetochore? A kinetochore is a sequence of DNA and some protein. Okay? It's a sequence of DNA and some protein, and each sister chromatid has a kinetochore. So, if this if this is a sister chromatid, I'm going to draw something right here and color it in, call that a kinetochore. And this sister chromatid has a kinetochore as well. Okay? Well, what happens with the kinetochore is some, not all, some spindle fibers will attach to the kinetochore. Okay? And these spindle fibers are coming from opposite poles of the cell. Okay? Opposite the centrosomes that are located at the opposite end. So let's go ahead and take a look at that right here. If you look, you can see the kinetochore, the dark spot on each sister chromatid, and you can see spindle fibers attaching to the kinetochore. Okay? So this one, for instance, has a spindle fiber from this centrosome and from this centrosome here. Okay? And this is just to show you again that spindle fibers will attach to the kinetochores. Okay? Now, that at this point should be, it should be start to clear things up as to how these sister chromatids will separate. You can kind of see that these spindle fibers will pull them apart, okay, and that will happen. Okay? Just a hint though, okay, so for prophase, this is all the things you would need to know. And on the exam, you know, I'll list some, if not all, of these events right here. And by each event, I'll have a blank line, and I will have a word bank with interphase, G1, S, G, and they won't be in order, G2, prophase, telophase, anaphase. And so if one of the events says chromatin condenses into observable chromosomes, and you have to put the right stage there, that would be prophase. Now, what is the best way to learn this? Because we're not done yet. We've still got a ways to go. What is the best way to learn this? Make note cards. Okay? On one side of the note card, put prophase. On the other side, put all the stages. And remember those stages. Okay? You remember those stages when you come across prophase. So you quiz yourself over and over again till you get it down. Okay? So now we're done with prophase. We're going to metaphase. So what happens during metaphase? Well, if you kind of remember with the beads and with some of the slides that I showed up here, metaphase, what's going to happen is the chromatid pairs are going to be moved by the spindle fibers, okay, to the center of the cell. The chromatid pairs will be moved by the spindle fibers to the center of the cell. Okay? And what's going to happen is the chromatid pairs will be lined up along the equator of the cell, and that's an imaginary line called the metaphase plate. Okay? So let's take a look at this. So here is metaphase right here. So these spindle fibers that are attached to the kinetochores moved the chromatids to the middle of the cell, to the metaphase plate, where with each pair, one chromatid is oriented toward one pole, and the other chromatid is oriented towards the other pole. Okay? And that's what happens during metaphase. So we still have chromatids at metaphase, sister chromatids. At anaphase, the sister chromatids separate. Once a pair of chromatids separate, each chromatid then becomes a full-fledged chromosome. Okay? So in anaphase, the sister chromatids separate and move along the kinetochore microtubules toward opposite ends of the cell. Okay? So the sister chromatids will separate and move along kinetochore microtubules toward the opposite ends of the cell. Okay? So the chromosomes no longer exist as chromatids. They are separated. Okay? So here is anaphase. So you can see what's happening here. These microtubules are basically yanking these chromatids toward opposite poles of the cell. When they're no longer chromatids, they are full-fledged chromosomes. So you can see that each cell is going to get an identical complement of DNA because these two chromosomes are genetically identical. One's going here, one's going here. These are identical. One's going here, one's going here. So on. Now we want to focus on how it is these chromosomes are moving. What has to be happening to these microtubules right here? Okay? The microtubules shorten by depolymerizing at the kinetochore ends. What does depolymerizing mean, do you think? Think of what polymerizing means. What polymerizing means basically making a polymer, and there's a process that makes a polymer. What is depolymerizing?
It's hydrolyzed at the end?
Yes, hydrolysis is occurring at the end. Depolymerizing, in other words, breaking it down. Okay? So the microtubules will shorten by depolymerizing at their kinetochore ends. All right? Now this, I don't care about this, this, or this. I want to focus on this bottom one here. This is showing what is happening at the kinetochore end. So here's a chromosome right here. Here's a microtubule. The centrosome is way over here, okay? And this microtubule is pulling this chromosome toward the centrosome, but the microtubule has to get shorter to do that. So what happens here? This motor protein just helps with the movement here, but there is hydrolysis that occurs here, and we've got a bunch of tubulin subunits that are hydrolyzed off. These aren't individual amino acids. These are subunits of that globular protein tubulin that we learned about, okay? But hydrolysis occurs here. Depolymerization, okay? By the way, there's a question in your lab that I kind of pointed out about a textbook hypothesis on how these chromosomes are being moved, talking about the spindle a little bit, and it's referring to this, okay? Something else happens during anaphase. Non-kinetochore microtubules from opposite poles, in other words, the parts of the spindle that did not attach to the kinetochore, from opposite poles, they will overlap and push against each other, elongating the cell. Okay? We're just showing some overlapping here. There's no elongating here. This is just a close view of overlapping. But I say this happens in anaphase. This is metaphase here. It doesn't. I say it happens in anaphase, so that's what you'll want to answer on the exam. Okay? So there's some overlapping of non-kinetochore microtubules. You have to get used to that language because that's what's used on the exam. Okay? So you can see it here. See how this cell has gotten longer? It's a little longer than this one. These non-kinetic or microtubules overlap, push against each other, and elongate the cell. Well, how are those things getting longer? If the other ones get shorter, if the kinetic or microtubules get shorter by depolymerizing, how are the other ones getting longer? Polymerizing, right. Dehydration synthesis of those tubulin subunits. Okay? So these are getting longer. So that's what you need to know for anaphase.
Can you go back to that last slide with the definition of the non-kinetic?
Non-kinetic or microtubules from opposite poles overlap and push against each other, elongating the cell. That one? And here they are, pushing against each other, elongating the cell. So all of those things happen during anaphase. Okay? So I could have this as one of the events right here, and I've got all of the stages of the cell cycle. Telophase, cytokinesis, okay? Prophase, metaphase, anaphase, and I'll have a line after this, and you look at the word bank, and oh, there's anaphase right there, and you put it in there, okay? Like I said, the best way, at least for me, to learn this would be making note cards. And the great thing about note cards is you learn it, you learn the stuff as you're doing it. You're 50% there after you make them. Now, if you buy note cards from somebody, that's good. Okay, you want to save some time, but you don't get the benefit of making them. Okay? You can still actively learn by purchasing note cards. There's nothing wrong with it. Okay, so now we have telophase. Telophase is the opposite of prophase. So if you know what is happening during prophase, then you just reverse it, and that's what happens during telophase. So for example, I could have as an event, the nuclear membrane reforms, and you would select telophase, okay? You would select telophase because during prophase, the nuclear membrane breaks down, okay? So learn prophase, and the opposite happens during telophase, okay? If the spindle forms during prophase, it breaks down during telophase. If the chromatin condenses during prophase, then it decondenses during telophase. So just learn all the events of prophase and apply the opposite to telophase. Okay? Now, cytokinesis occurs at the same time as prophase. They are not the same thing, though. I have students answer, oh, this is telophase. No, that's cytokinesis. You said they were the same thing. No, I didn't. I said they occur at the same time. They occur simultaneously, okay? So cytokinesis is the cytoplasmic division, all right? So here is telophase here. It's showing the nucleus. Here's the nucleolus reappearing here, okay? Here is the nucleus is starting to reform. Notice the spindle is gone, okay? And we'll get to this thing, this cleavage furrow, in a minute. So that represents cytokinesis when you see a cleavage furrow in animal cells, okay? So in animal cells, cytokinesis occurs by a process known as cleavage, forming a cleavage furrow, okay? So here it is right here. Here's an animal cell. And there's a cleavage furrow. Let's go down to the cartoon. Okay? A cleavage furrow forms right here. and what structures are actually forming that functioning cleavage furrow. A contractile ring of microfilaments. Yes, you need to know it to that level. Those aren't microtubules. Those are microfilaments that form the cleavage furrow. Okay? A contractile ring of microfilaments, and that's going to work like a drawstring, and it's going to contract and contract and contract until the cell pinches into two cells. Okay? Now, plants' cytokinesis is different, and you did see this a little bit on the onion slides, okay, the onion cells there. In plant cells, instead of a cleavage furrow, a cell plate forms during cytokinesis. So here is showing the cell plate. What is it actually? What is the cell plate? There are vesicles that come and line up along what used to be the metaphase plate. And those vesicles carry a lot of cell wall building material. What's the main component of plant cell walls? Cellulose. So there will be cellulose in these vesicles, and these vesicles will grow out and fuse with each other, forming the cell plate. And then the cell plate will extend out and fuse with the cell membrane and the cell wall and become the new plant cell wall. Now, you may say, well, this plant cell was twice the size of these two new ones. And I would say you're right. But what will happen many times is during cell division, these cells, the cell walls will remain pliable and the cells can grow some, okay, until they get to a fixed size. Okay? And here's the onion that you saw earlier, not the same slides. These won't be on the lab exam, but this is just showing you, I mean, if you looked under the microscopes, you may have seen some cells like that, okay? So there's your prophase, and that's still prophase. There's metaphase, anaphase, and telophase with cytokinesis. So you see the cell plate forming there. Now, I want to show you a video on mitosis, and then I want to look at today's lab a little bit. So let's see this video first. So we're actually, let me put this back up to here, cytokinesis. So all of those events that happened, a good number of those will be included, 18 of them to be exact, okay? Will be included on that section on exam 2. Isn't it weird to be talking about exam 2 already when you just took exam 1? And it was a long test, I know. It's actually 1.5 exams, okay? All right, so let's see. Mytosis. Here we go. Oh.
This soccer player's knee will be as good as new in a few weeks.
Hang on. I don't know. I don't play soccer. I never did. Football, wrestling. Is that an injury in soccer? Okay, all right. Just, I do have to say though, soccer is a legitimately rough sport. The sport with the most concussions, bad concussions, would be women's soccer at the collegiate level and pro level. They have more concussions, the higher rate of serious concussions than football. Yep, women's soccer, not men's, women's.
Thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers.
Look at, even they include prometaphase.
Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if